A very good evening from me and also Mr. Gregor Johann Mendel, who is my dearest friend out there, my dear children. We heard about his story, how he never gave up, just like you guys, okay? How he was always a fighter, how he worked on garden peas experiment for almost 7 years of his lifetime, my dear children. But then, just like any of us, in his story also, there was a big twist. And that twist obviously was not that great. Okay, so there were people who questioned like, did Mendel go wrong somewhere? Should we take away the title of father of modern genetics from him? And all these questions around it, just like how it happens in your life, my dear children. So at one point you might be doing really good in academics, and then all of a sudden probably you might be distracted, or you might be not well, or you might be not working hard. People start questioning, oh, your marks are not that great. So there's something that is going wrong. Similarly, Mendel also faced this particular thing. But why did he face this big question in his life after doing an experiment for 7 long years? So the problem with Mendel was, he said that whatever gene that you're taking, my dear children, this gene, for example, the gene is there for height. He said this gene can have 2 alleles, allele 1 and allele 2. Okay, allele 1 would be coding for tallness and allele 2 would be coding for what? Dwarfness. This was the thing that he said. And he also said that one allele will always be dominant. What do you mean by that? Basically, for that you need to understand the structure of the chromosome. So this is a chromosome, my dear children, and over here suppose I have a gene coding for a protein which is very much important for producing growth hormones. Now if this protein is there, growth hormone will be there, and which is why what will happen, suppose there is enough amount of protein to make enough amount of growth hormone, this organism which is having this particular chromosome might be tall. Okay, so this is one scenario. Now there can be another scenario also where, my dear children, another chromosome, same place, a gene is there, but this gene does not code for very much amount of protein, it's very less protein. And this very less protein cannot make much amount of growth hormone. So obviously, no much height would be there. If no growth hormone, no much height will be there. So this allele or this gene is coding for what? Dwarfness. So I see 2 types of gene coding for the same character, height. This is tall, this is dwarf. Both of them is what? We are talking about height itself, right? So when we have 2 genes coding for 2 contrasting traits, we call them what? Alleles. We call them alleles. And always remember, you get one chromosome from your parent, another chromosome from another parent, okay? And at the same location, you would have a gene for what? Height. Suppose here you have the allele small t and here you have the allele capital T. Okay, here you have the allele capital T. That means here there is a protein that can produce enough amount of growth hormone, and there we have a protein that does not produce enough amount of growth hormone, my dear children. What will happen? Will that person be tall or dwarf? Here anyway, there is a protein there is a gene that is coding for good amount of protein, which is why, my dear children, it will be dominating over this, and the height will be what? Tall. It will be just tall. So this is what he said, that there are only 2 alleles of a gene. For whatever gene that you're talking about, whether it be height, whether it be shape of the seed, whatever is there can be 2 things. There can be 2 alleles, and one allele will be always dominant over the another one. So he said, suppose, forget about the tallness and dwarfness and stuff like that. Let's talk about the flowers. I have a red flower, I have a white flower of the same plant. Okay, the same plant have 2 different type of flowers. Suppose I have a red and a white. Okay, 2 different plant of the same species, I'm crossing them. I would be able to see that the progeny, the new baby plants will be having red flowers. This is what he said, because according to him, the red color thing was what? Dominant over the white one. So this is what he had in his mind. But then there was a twist, as I said. There were people who did experiments, who saw things, and I said that, but that's not the case, sir. There is a third color. So when people actually cross between the red flower plant and the white flower plant, they were able to get a all new color which is not clearly visible over here. That was pink. That was pink. But if this was totally dominant over this, then probably it should have been just red. Why is there a pink thing? Why is there a third color? That means Mendel did something wrong. All his findings were not exactly correct. That is what we get to understand, right? But for that, we need to do our experiment. And in this video, we are going to do that experiment and see who is going to win. Mendel or the other guys. Sorry, Mendel or the other guys out there. And I want one thing from you in this particular lecture. Okay, and that is a challenge. The challenge is, from this point onwards, until the lecture finishes, you have to be with me. Whether you are watching it, or in the previous session, or probably you're watching it at night, make sure you're watching the complete video, because I am going to make sure I teach you each and every single thing that could take away all of your doubts from this particular chapter. Make sure you take up that challenge. If you're accepting the challenge, write down in the comment section right now, challenge accepted. And I'm telling you, let me see who wins, you or me. So not everyone can take up this challenge. It's not very easy to study a whole, whole different set of topics in one particular lecture. But if you think you can make it, accept this challenge and go for it. Let's see who all are able to understand. And how will I understand whether you are understanding things or not? By you answering the questions that I am asking in the session. Okay, there will be certain homework question, you have to answer it as well. Deal? I hope that's a deal, and let's go ahead. Children, the only reason why I'm asking you to take up that challenge is because it's very important for you to study this chapter. It carries a huge lot of marks, 13 marks in your PU2 and 4 to 5 marks in your KCET, a chapter that has the most highest weightage in KCET. So make sure, and also in your PU2. Okay, so in PU2, molecular basis of inheritance and principles of inheritance are something that you should never miss. Okay, they might seem difficult, they might seem new, but it's not at all difficult. It's easy. It's just that there are so many new things. Okay, so let's get ahead. And over here in today's session, we are going to talk about monohybrid cross, dominance, codominance, multiple allelism, pleiotropy, and polygyny. So some people told me, ma'am, our portions are until pleiotropy or polygyny. So I thought I can include these two topics here itself because that makes sense. Okay, not going just according to the NCERT. Until multiple allelism, we will be going according to NCERT, and then have taken pleiotropy and polygyny, because you will understand it better if I teach you right here itself. We have dihybrid cross and we have linkage and recombination. I will be doing it in the next session. Okay, so that's deal. And if at all you want the PDF, what you have to do, please join our WhatsApp channel. You'll get links to download the PDF. Now, talking about the winners that I have for the previous session, kudos to you. I actually found so many people out there participating in this, and I'm truly, genuinely, whatever you say. Okay, I don't know how to put it down together, but I'm so happy that you're taking everything seriously. You're participating really well, and I'm super proud of you. Okay, maybe Prajwal, who had given me all the answers, homework number 1, homework number 2, 3, and then we have Manvita Reddy also again with all the answers. By seeing this, I understand that whatever I'm teaching, you're getting that. Okay, so kudos to you. And also Manasa. Okay, Manasa has been putting down the comments regarding all the answers that is there, my dear children. Good, very good. Okay, super proud. Even Sagar out there. So Sagar have given me answer for all the question along with the timings. Awesome. And Prajwal said that you're teaching, the graph is up. So thank you so much. And I've also commented back one very important thing, that is your hard work is also graph up. So it's the thing that I can see, my dear children. Please put in your hard work, and I'm pretty sure you are gonna get everything that you're looking forward for. Okay, so accepting the challenge, we are getting ahead with the chapter. And do not forget to put down the homework answers. Okay, and do not forget to hit the like button if at all you're liking this session, and please stay motivated. We need a little motivation in studying this chapter. Okay, so the first thing that we are gonna go ahead is Mendel's experiment on peace. Do you remember the dates? Do you remember the years that he worked? It was 1856 to 1863. This is an important MCQ question. For 7 years he worked. The years were 1856 to 1863. Always remember that. Okay. Now, why did people believe his work? Because he was the first one to use math in biology. We do not like it, but yes, that's how he did it. So Mendel was the first to apply statistical analysis and mathematic logical study inheritance. For example, if I just go, these are my studies, please look at it. You won't believe it, okay? I should have some evidence, I should have some numbers. Then only probably people could believe it. So he made science more logical, more scientific, okay? So that was one very important thing. Second thing, he had a large sample size. He was not like that, I did experiment on 1 or 2 things and that's all. No, he experimented on thousands of tree plants, my dear children, which made his results what? More reliable and accurate. People believe you more then. Then he did repeated experiments. It's not like 1 or 3 experiments, that's all. No. For 7 years, my dear children, he continuously did that, saw that, was there a variation from what he was observing? So he repeated his crosses over several generations, proving that his results were consistent and not what? Random out there. And fourth thing, he chose clear and contrasting traits. Whenever he did his experiments, he made sure he was using 2 contrasting traits out there. For example, it's easy for him to observe. You cannot see a genetic makeup of a plant out there, right? I see a tall plant. How do I know the genetic makeup? What are the genes in that? No, I cannot know that. So basically, for understanding things better, he used traits like tall versus dwarf, yellow versus green and things like that, which were easy to observe and compare. So these were the reasons why people actually believed him. First, use of math, large sample size, repeated experiments and usage of clear and contrasting traits for the experiment. Okay? Now, going ahead with the next question, that is, I said that he used garden pea. Okay, he used what? Garden pea. In the initial part of the chapter, I said that he worked on garden pea. Please remember this particular name also. Okay, but why did he choose that? Here you get a clue, because it had contrasting traits. But how many? That's the question. So this again is an important PYQ. Okay, so there were 7 pair of contrasting characters. 7 pair. Okay, 1 pair contains how many things? 2 things, right? 1 pair means 2 things. So 7 pair means 7 into 2, 12. 7 into 2, 12. So 7 pair of contrasting characters means how many traits in total? I told you, whenever we say character, so suppose character can be height. Now the sub part of the height can be tall, height can be dwarf. So what do I have? What do I call them? I call them trait. I call them trait. So I have a character, height, and in that I have 2 traits. So this is a pair of trait that I observe. Okay, so pair of contrasting characters, or how many traits in total? If you are asking about traits, it's almost 12 of them. 14 of them. How many? 14 of them. You will understand this better. If at all you are not understanding it now, please don't worry. Listen to me. Okay, so over here, he chose flower color. Flower color. This was a character. In that you had 2 traits. Flower color could be purple or violet, my dear children. Please write violet. Or it could be white. Okay. This was dominant and this was recessive. How did he come to know? By doing the experiments. Okay. He took the flower color. Now I'll go to flower position. Okay. So flower color and then flower position. Flower position, 2 type of things he saw. One was axial and the other one was terminal. What do you mean by axial? For example, this is a plant. Now from where you get the leaves, suppose there are flowers over here. So you call this as what? Axial position of flowering. Terminal position means, for example, there is a branch over here, there's flower over here. Suppose it goes and here the flower is there. You call this as terminal position. Now which one was dominant? This one was dominant. Okay, axial positioning was dominant. So flower color is one particular character. Flower position is another particular character. So you have got 2 characters. Okay, 2 characters with 1 pair of traits with another pair of traits. Fine. So we have done 4. Now going ahead, flower color, flower position is done. Now let's go with seed color and seed shape. Okay, seed color, there were 2, yellow and then green. So usually when we have peas, we have green color peas, no? Right? But that is not the actual dominant one. Always remember, it's yellow. Yellow is the one which is dominant. So yellow seed color is dominant, whereas green is what? Recessive trait. Now seed shape. This is the next character, 4th character. Seed shape, round, that is dominant, wrinkled. Okay, so the surface, if it is like this, it will be wrinkled. So that is what? Recessive character. So my dear children, how many have we covered? Flower color. Violet is the one that is dominant. Violet is the color that is dominant, right? It is clearly visible. You can remember like that. Okay. And white is recessive. Flower position is there. You have axial and you have terminal. Axial one is the dominant. A comes first. So that is dominant. You can remember like that. Now going ahead, with what? Seed color. Seed color, the green ones are the ones we eat, but it should be actually yellow. Yellow are the dominant one. So yellow is the dominant and green is the recessive one. Seed shape, round is the dominant, and my dear children, wrinkled is the recessive one. That is done. Now we have pod color and pod shape. What do you mean by pod? My dear children, green peas are usually inside this particular covering, right? So this is known as pod. So what is the pod color? Usually when we take green peas, do you go and search for yellow ones? No. There the twist is there. There's the green which is dominant, and the yellow one which is there, it would be the recessive one. Okay, so you always pick for the green ones, right? So you pick that because that's the dominant one. You can remember like that. So pod color, green is dominant and yellow is recessive. Pod shape, my dear children, if you talk about that, full as in it's nice only. Okay, it's not dry like this. Suppose it's dry like this, my dear children, this will be known as constricted. This is known as constricted. Okay, full means it's proper. It's proper like this. Okay, so inflated or full, either words we use, inflated or full, that is a pod shape which is dominant. And over here, constricted one, or what do you call, constricted or flat. Constricted is a word that you use, that is a recessive one. So that is pod color and pod shape. So why is this important? Because you get questions from this. Okay, that is why this is very, very, very important. You will be asked, what is that character that he didn't study? For that, you should know what is the character that he did study. Right? So my dear children, always remember, let's start with flowers. Okay, flower color, okay, flower position, fine, flower color, flower position. Now talking about seed color, okay, there will be seed. So flower gives the seed, my dear children. So seed color and then seed shape. Similarly, my dear children, the seeds are inside the pod. So the pod color and the pod shape. And over here you have one more very important thing, that is plant height. That is the one that is mostly discussed. So that is tall, and here you have dwarf or short also. So these were the 7 characters that he studied. If you take flower together, okay, flower color, flower position, seed color, seed shape, pod color, pod shape, and then plant height, then in that case you will be able to remember things better. Remember 7 pair of characters are there, and or what you call 7 pairs of contrasting traits are there. You can see like that. 7 pairs of contrasting traits are there, and how many in total? If you're asked, 7 into 2, 14 of them in total. Always remember that. Or you can say 7 contrasting characters, and in that you have 14 traits out there. That would also be best. I hope this part of the story is clear to you. Okay, so what all things are clear? The things that are clear is why did people believe him, or why were his experiments really good. Okay, what was the year? 1856 to 1863. He worked on garden pea for how many years? 7 years. Okay, these are all questions. How many characters were there? 7 pair of contrasting characters, or what do you call? 7 characters were there. In that you had 2 of them a pair. So basically 14 traits were studied. 14 traits were studied. 7 characters, 14 traits. Okay, now going ahead with the next thing that is out there. Ever wondered how passing of genes work? So when I say tall and this comes and then the plant becomes tall, do you know how it actually works? So I taught this in the basics, okay, in the previous session, but still I'm telling you, I won't take much of your time. So over here I have a tall plant, I have a dwarf plant. Okay, I have a tall plant, I have a dwarf plant. What happens is that this plant will make gametes. This plant will also make gametes. This was the homework question. Whenever they make gametes, they undergo what division? They undergo meiosis or reductional division, my dear children. Now when you call reductional division, that means that if you have a pair of chromosome, only one would be coming in the gametes. Only one can participate in the gametes, either this or this. Okay, so suppose here capital T came in, and from here small t came in. Okay, in the gametes. Now both these gametes fuses, I get again a pair back. I get again a pair back. Over here I have capital T, and over here I have small t. In this case, the plant which is having this would be what? Tall or dwarf? It has the dominant allele. Why do I call it dominant? Because it is able to produce more amount of proteins. It is more amount of proteins, so it will show that up, right? So that is why this is dominant. So my dear children, this plant will be what? Tall. So this is how things work. Now over here, I want to tell you one very important thing. When the condition is like this, when both the alleles are same, capital T, you call this condition as homozygous condition. Homozygous condition. Same thing goes here also, small t, is it homozygous or not? Yes, it is homozygous condition. Cool. So in this homozygote condition, whatever gametes you make, my dear children, from this plant, it will always carry capital T, and here also it will always carry small t. Heterozygous is a condition, my dear children, where you have capital T and small t. Now here if you want to make gametes, some gametes will contain capital T, some gametes will contain small t. Always remember. And one very important thing, the genes coding for the same character will be in the same location, which is why it is highlighted like this. Now we are going ahead with what? Jasmine, please talk about F1, F2 generation ratio, etc. So we are going to talk about the experiment that was done by Mendel, and please listen to me carefully. So what he did was, he took two plants, okay, one was tall and the other one was dwarf. The important character that you have to remember is that both of them were homozygous, or you also call that as pure breed. What do you call? Pure breed or homozygous? If you still didn't understand, I'll tell you. So both of them, when you talk about tall, you have to write like this: parents. Tall, that means it is capital T, capital T. Dwarf means it is small t, small t. Since they are homozygous or pure breed, they are like this. Both the alleles are same. These are alleles and both of them are same over here. Now, talking about the gametes that are going to be formed. In exam, you just write like this, okay? So capital T can be in a gamete. This chromosome can also be in a gamete. Here small t can be in a gamete, or this can also be in a gamete. We never know. Okay, we never know. These are the chances of gametes being formed. Now, the first generation that you're gonna get, okay, you call this as F1 generation. What do you call? F1, where F stands for filial. F stands for filial. So how do you cross it? Listen, let's see. So suppose this chromosome this gamete fuses with this gamete. In that case, what will you get? Capital T, small t, ma'am. Suppose this one fuses, this one fuses with this. In that case also, what will I get? Capital T, small t. Suppose this one fuses with this. In that case, again capital T, small t. Here, this one fuses with this, I get capital T, small t. Now, my dear children, tell me, are these progeny, so this is F1 generation that we are getting, are these progenies heterozygous or homozygous? Heterozygous or homozygous? They are hetero. Why? Because they have two different alleles, capital T, small t, capital T, small t. So, I can write one very important thing that I got to know is that they are all heterozygous. And what is that character? What is that trait? Are they tall or dwarf? They would be tall. Why? Because you have a dominant allele over here. This will be dominating. So I can say they are all heterozygous tall. They are all heterozygous tall. But Mendel didn't know this part of the story. Mendel was unsure about this. He didn't know. He cannot see the genetics, right? He was just doing the experiment. He just found that all the plants were tall. That means he was able to understand the phenotype, the physical appearance. That is, all of them were tall. He was not able to understand the genetic thing. So for understanding the genetic thing, for understanding the genetic makeup, he thought I should cross these plants. I should cross these plants, and probably I will be able to get it. So now these guys are going to be the parents. The F1 plants are going to be the parents. So parents, I'll write it over here again. And what do I have? I have capital T, small t. And again I have capital T, small t. So this is a tall parent. This is also a tall parent. I do not know what is the genetic makeup. Now, gametes will be formed. My dear children, gametes can be the chromosome containing capital T can go in the gamete, or the chromosome containing small t can go. Similarly, over here, chromosome containing capital T can go, or chromosome containing small t can go. Agreed? Okay. Now, over here, the next generation that I'm going to get, what do you call this? F1 generation again? No, this is F2 generation, filial 2 generation. So this is F2 generation, and let's cross. Always remember, cross like this, 1, and it goes like this. So it is capital T, capital T. Now this one here itself go like that, under this, okay. So that will make it look clean. So capital T, small t. Now this one, it goes with this. Always write the dominant character first, capital T, small t. Okay, this is the dominant character, I can see that. And this, with this I get small t, small t. And then Mendel was surprised, because he saw one thing which he didn't see here, that is dwarf plants. Where are the dwarf plants? Here, my dear children, when you see this small t, isn't it dwarf? Okay, so recessive characters would be showing them up when there is no dominant character. There is no one to dominate these guys out there. So my dear children, this plant will be what? Dwarf. This plant will be dwarf. What about these three plants? This is for short, tall. What about them? This is also tall because there is one to dominate over here. So three of them, my dear children, if you see, they were tall. They were tall. Three of them tall and one of them dwarf. Now, I am putting the term ratio. If you want to tell the ratio, the phenotypic ratio, the physical appearance ratio. Ma'am, three of them were tall and one of them were dwarf. So, the ratio is 3:1. You always write the dominant character first, like this, when it comes like this. 3:1 is what? The phenotypic ratio. Ma'am, please tell about genotypic ratio. What do you mean by genotype? Genotype means genetic makeup. So, what is the genotypic makeup? So, over here I have one with capital T, homozygous condition. Okay, so that is 1. So, I will write 1 and then capital T, Okay, I have 2 of them with capital T, small t. So, 2, capital T, small t. And then I have one of them with small t. So, 1 with small t. So, 1:2:1 is what? The genotypic ratio. What is the phenotypic ratio? 3:1. 3 of them are tall, 1 is dwarf. I hope you get it. I hope you get it. So, over here you do not need to talk about ratio in F1 generation because you don't see that difference. All of them are tall. Okay, and all of them have the same genotype. But over here you have to talk about the genotypic and phenotypic ratio. F2 generation will always have 3:1 ratio if the parents are like this. Okay, if only the parents are like this. That is a phenotypic ratio. Okay, so your homework question right now. This is the first homework question. Tell me the generation, what do you call? Tell me the phenotypic and genotypic ratio of F2 generation in this case. I've written it already, but I want you to write in the comment section and tell me, because that is how you remember and you have to clearly tell me. Okay, this is your homework number 1 question. So I suppose this is understood and this was his experiment. He was able to find, okay, as of now we know there is dominant allele, recessive allele and things like that, but for him it was all new. Right, at that particular time he said that I crossed between 2 plants, tall and dwarf, and all of them were tall. And the second generation I was able to see what? The dwarf plant out there. That means probably here also the gene was there, the dwarf gene was there, but something was dominating over that. Okay, and now it's time for understanding what he found from these experiments out there, my dear children. So there is a question out there. I'll make you solve this question. Oh, you saw that answer? Okay, wait. Where is it? Okay, this were the findings. Okay, so Mendel proposed that something is stably passed down from the parent to the offsprings through the gametes over generations. What was that something? He said that something was passed from the parents to the progeny out there through the gametes. What were these things? These were factors, or what do you call this as now? You call that as genes, my dear children. So from the parents have a chromosome, parents will have the gene, this gene will go to the gamete, okay, this whole chromosome goes to the gamete, and from the gamete it passes to the progeny, my dear children. So what is this particular thing which is usually being passed down? This is nothing but the factor. According to him it was factor, according to us it is what? Gene, my dear children. So that was first thing that he said. The second thing, genes are units of inheritance, that we are saying, okay. Genes are what? Units of inheritance. That is the only thing that is allowing us to inherit stuffs. So genes carry information required to express specific traits in an organism. What information does it carry, my dear children? It carries the information as to what protein has to be made. And this protein, my dear children, probably might be helping in making A to B. And this B might be helping in giving a character. Get it? So proteins are what? Like a midway, a friend, middle friend that you have. Okay, and genes do that. Genes do what? Genes basically helps in coding this protein. Okay, genes carry the information as to what protein has to be made. A bad protein, a good protein, defective protein, whatever is there. So genes are units of inheritance and genes carry the information required to express the specific traits. He didn't say this, it's said by us. Okay, so first thing that he said was this, that all the there's something that is stably passed down from generation to generation, and that is what you call as factors. Now we know that it is genes. Now he also said that alleles are different forms of the same factor. There are some things, okay, there are some alleles that you see, and these are nothing but different forms of the same gene. So same gene as I told you, there is gene for height. So there is gene 1, there is gene 2. Suppose this is coding for less amount of protein and this is coding for more amount of protein. So I'll call this as allele 1 and allele 2. Can I call them same? No, they are different. They are different things, but they come under the same category of genes coding for what? Height. So my dear children, he said the alleles are different forms of same gene that code for a pair of contrasting traits. That code for what? A pair of contrasting traits. Yes, he proposed that in a pair of dissimilar factors, one dominates the other. When there is a pair of dissimilar factors, suppose there is T and capital T, one is going to dominate over the other one. The one that dominates is known as dominant allele or the dominant factor or the dominant gene, and this will be known as what? The recessive allele or the recessive factor or the recessive gene. Gene term is used by us. I am telling you again. He also said that during meiosis, allele pairs segregate and only one is passed to each gamete. Now, it might be too difficult for you to understand or decode this, but it is so easy. So, my dear children, he is saying, suppose this is a parent 1 and this is parent 2. So, parent 1 will be having a pair of chromosome, parent 2 will also be having a pair of chromosomes. Suppose it is capital T and here it is small t, suppose here it is capital T, here it is small t. He said that when you form the gametes, okay, when you form the gametes, what is going to happen? These two chromosomes are going to segregate. These two chromosomes are going to segregate. But did he use the term chromosomes? No. He said that these two factors which are there, they are going to separate. They are going to segregate and only one of them will be going to each gamete. Isn't that correct? Yes. Because during meiosis, half of it will be going. Either this can go or probably this can go. So, that's written over here. He also said during meiosis, allele pairs or the factors that you see there, they segregate and only one is passed down to each gamete. From here also you get the same thing, and that's what we write, capital T and small t. It's just the chromosomes which are passing down or the genes which are passing down, my dear children. And when you cross this and this, you get your thing. What is the genotype of the progeny plant? That is the thing. So, I know it's too difficult to take all of this in. I'll repeat it once more. Listen to me carefully. And my dear children, if you are understanding, do not forget to click on the like button. Okay. So, that is, Mendel said that all the things that you see today, okay, or you see in the progeny, it's passed down generation from generation in the form of something that you call as factors. And now these factors that you see can be of two different types, can be of a pair of contrasting traits. Okay. Now, one of them will be dominant and the other one will be recessive. That is the third thing that he said. Now, the fourth thing that he said that basically during gamete formation, it's not like both the chromosome or both the factors would be coming together. Only one of them would be coming. How is that happening? Because they segregate or they separate before the gamete formation. So, that is being written over here. Now, whatever was his findings, he put down laws out of it. How? He clubbed all of these points together from here to here into one law and from here to here into another law. And what are the names of these laws? The first law that you see is law of dominance, where he has put down 3 points. Characters are controlled by discrete units called factors, isn't it? Yes, characters that you see are controlled by discrete units known as factors, which you call as genes. Now, the factors occurs in pair, genes occurs in pair, right? That is known as alleles. And in a dissimilar pair of factors, in a dissimilar pair of alleles, one member of the pair dominates over the other one, which is recessive. 3 things clear? Character controlled by factors, factors occurs in pairs, one would be dominant, other would be recessive. Okay. I have put the exact word that is given in your NCERT. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross. So, this cross that we just studied, my dear children, it's a monohybrid cross. Why? Because we are studying about just one character, that is height. And in that we are creating a hybrid. Tall plant, dwarf plant comes together, I get capital T, small t. This is a hybrid that I am forming. So, this is what monohybrid cross that I am having. And the law of dominance is helping me to explain that why only one character is being seen in the case when things are like this, right? Because the one is dominating over the other one. That's the reason. So I have got a reason to understand why is it like this. Why in F1 generation there is no dwarf plants? Why in F2 generation only there is dwarf plants? I am able to understand that. So law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in F1 generation and the expression of both in the F2 generation. It also explains the proportion of 3:1 obtained at the F2 generation. So three things are explained by law of dominance. Why? One, why does only one thing express in F1 generation? Second thing, why both of them reappear in F2 generation, and why is the ratio 3:1 obtained in F2 generation? What is 3:1 ratio? It is the phenotypic ratio, remember that. So that is law of dominance. He said three things and then he said that law of dominance is used to explain all of this thing. Okay, got it? Characters occur in pair. Sorry, characters are maintained by factors occurs in pair, one dominates over the other, and this thing. That's all. That is law of dominance. Now the second thing that he put down was law of segregation. I told you in segregation, what did I tell you? That there is a pair of factors out there, they will segregate and only one will be going to one gamete. So he said alleles do not blend, both reappear unchanged in F2 generation. That is, there is no blending that is being seen. Okay, this and this remain separate. You do not see any blending out there. And basically they would reappear unchanged in F2 generation. If there was blending, my dear children, if both of them blended somewhere, okay, my dear children, would you be able to see the dwarf character in F2 generation? You won't be able to see that because it would have been blended, but that's not the case. Alleles do not blend, both reappear unchanged in F2 generation. Now second thing, during gamete formation, allele pairs, the pair of alleles, they separate. So each gamete gets only one allele. So each gamete which is going to be formed, it will get only one allele. It's very easy if you understand the logic behind it. There is a pair of alleles during gamete generation, only one allele will be passing in a gamete. That's all. Homozygous parents produced identical gametes. Yes, homozygous parents, as in, for example, capital T, capital T. What gametes will it produce? It will produce the same type of gametes. Can be capital T, that is written over here. Homozygous parents produce identical gametes, while heterozygous will be producing dissimilar or two type of gametes. So this can produce small t or it can produce capital T. What is the chances of this producing capital T? 50%. What is the chance of this producing small t? 50%. Okay, so produce two type of the ones 50% each. So law of segregation, do not think that you have so much things to remember. Okay, I told you these points that you see over here in the first part, okay, it has been broken down into two things. So all his initial findings, that chromosome, factors in pair, one dominating and all those things in one law. The second one where they are separating, that will be separate law. So that is law of segregation. In law of segregation, alleles do not blend, they remain unchanged. You can see that alleles come back in F2 generation. And then he said during gamete formation, only one will be going. And my dear children, if it is a homozygous condition, homozygous parent is there, then you will produce same type of gametes, identical gametes. If there is heterozygous parent, you produce two type of gametes out there. Got it? That's all. That's the law of segregation. Ma'am, is there a third law? Yes, there is a third law. But you can only study it after you study dihybrid cross. We'll study it later. Okay, we will study it later. So I hope you have understood the findings. Okay, you have understood the findings and how it was put down in the form of laws as well. Is that clear? I hope it's clear. If you have doubt, my dear children, it's totally okay to have doubts. Please put it down in the comment section. I am here to sort it out. Okay, if it's all clear, then you have to tell me, ma'am, it's all clear. Ma'am, I am understanding things. Thank you for teaching. Whatever you want to say, you can say. If you're not understanding things, you can say like that also. But you have to make sure that you do not give up over here. Now, my dear children, I will be going back to the question that is there. Okay, so this is one question that I wanted you to practice. You will get these type of questions out there. So in one of the hybridization experiments, a homozygous dominant parent and a homozygous recessive parent are crossed for a trait. Plant shows Mendelian inheritance pattern. Okay, so what did he say? He said that, or what this particular question says, that there is a homozygous dominant parent and homozygous recessive parent. Example? You need to have an example to get the answer for this, no? Homozygous means same. Dominant means the dominant allele. So if I put like this, capital T, doesn't it matches the condition? They are homozygous and they are dominant also. And over here I have homozygous recessive one. So this is homozygous recessive one. Okay, if the question was heterozygous dominant, then how would have you written? You would have written like this, heterozygous and the dominant one. Got it? So here homozygous dominant and homozygous recessive has been asked, so we don't need to do that. Now we have to tell what is going to be seen. So dominant parental trait appears in F2 generation and recessive parent trait appears only in F1 generation. Do you think after crossing them in F1 generation you will see only the recessive trait? Do you see dwarf plants? Do you see dwarf plants? We have made it right over here. In F1 generation, when I cross them, my dear children, were you able to see any dwarf plant or recessive trait over here? No. So this statement is absolutely wrong. This statement is absolutely wrong when they say that dominant parent trait appears in F2 generation. No, dominant parent trait appears in F1 generation. It is dominant, so it has to appear in F1 generation. And recessive parent trait appears only in F1 generation. Wrong. This is wrong statement because here F1 and F2 is being written. If it had been reversed, then it would have been correct. So dominant parental trait appears in F1 generation. Is it? Do you see dominant parent trait in F1 generation? Yes. Okay. Now the second part, and recessive parent trait appears in F2 generation. Yes, Mendel thought it's not there, but it came back again in F2 generation. Yes, my dear children. So don't you think this is a correct statement? Yeah. Now dominant parent trait appears in both F1 and F2 generation and recessive parent trait appears only in F2 generation. Is that correct? I didn't see this option, okay. So see, me being a teacher, okay, even I went ahead and just, okay, this is the correct option, I went ahead with that. But now you have an option. Dominant parent trait appears in both F1 and F2 generation. Do you see them? Yes. In F1 and F2 generation, I can see, this is, here you have tall plants, here also you have tall plants, F1 and F2. But the recessive ones, the recessive parent trait appears only in F2 generation. Do you see it over here? No, you will see it only there. So this is again a correct statement. Okay, now dominant parent traits appear in F1 generation and recessive parent trait appears in F1 and F2 generation. Wrong. So my dear children, your answer would be this. Your answer would be this. So in the question I have marked this is not the one, this is the one. So the problem, see, I was preparing the PPT, I didn't have time, so I just came around here, gave the first answer, I thought this is the one. But now when I was explaining to you, I saw that there is another option. Same thing can happen to you in exam time also. Okay, you might be in hurry-burry. You might be in hurry-burry and you might be like going like this, and then you will see the first option seems correct and you might just go tick that, but then you have other option, which is why it is said you please read all the options out there. Okay, so that is one learning that you got from my mistake. So all the mistakes that you do in your life, you will have a learning, and this is today's learning of Jasmine Ma'am. That is, please Jasmine Ma'am, read the question carefully. The same thing probably you should not do, that's why I said that. Now, there is a homework question for the champions out there, because whatever I explained, if I think that you can do that question, I give you that question. Okay, so which among the following characters selected by Mendel in a pea plant is recessive character? Which among the following characters selected by the Mendel in the pea plant is a recessive character? Inflated pot, green pot color, white color, axillary flower. You can tell me in the comment section. This is question number 2, homework question number 2. Homework question number 1 was telling the phenotypic and genotypic ratio of F1 generation. Remember that. After putting that, you can put this answer, okay? And we studied the Mendel's law of inheritance as well. Right, what is left behind? Okay. So is there an easy way to cross? Is there an easy way to cross by doing this, apart from doing all this crossing things? The answer is, he says, yes bro. So I am just decoding what he's trying to say, okay? So yes bro, my name is Reginald C. Punnett. I am a British scientist and I know that's too much information for you to remember. So I am the one who put down the Punnett square, my dear children. What did he give down? He put down the Punnett square out there. And how does Punnett square help in what do you call crossing? So basically, suppose you have two plants, capital T, small t, capital T, small t. You want to cross them, you can do like this, like this. But that's too, what do you call, difficult when, yeah, that's too difficult sometimes. So what do? You make a Punnett square like this. Okay, you make 4 columns like this. Fine. And now write the one parent's gametes. So gametes can be either capital T or small t. Agreed? Here also gametes can be either capital T or small t. Agreed? Now suppose I cross this and this, what will I get? This plus this I get capital T small, capital T, capital T. This and this I get capital T small t. This and this I get capital T small t. This and this I get small t small t. Please see this. Please see this, you will understand it better. I crossed here and here, I got this. I crossed this and this, I got this. This and this, I got this. This and this, I got this. I got my answer. So 1, 2, 3 are tall, 1 is 2 off. I can cross it like this also. Okay, now a homework question to you. This is a question that I have just made right now. I want to cross I want to cross these question, homework number 3. Okay, capital T and capital T small t. What is going to be the phenotypic and genotypic ratio? Phenotypic and genotypic ratio. Tell me. This is the homework question number 3, and sometimes I only forget after giving you homework what was the question. So I'm just gonna take a picture. Done. Okay. So you have to tell me the answer. I have the picture, I'll check it up. So please do that and let me know. You can use Punnett square for that this time. Fine. Phenotypic and genotypic ratio once you cross this. Cool, bro. Now, ma'am, I have a doubt. That's you. Okay, that's you guys. It can be a girl or a boy. I just found this picture, so I just put it. Okay, so how did Mendel get to know if the plant is tall? Like this, capital T or capital T small t. For example, I see a plant. Okay, I see a plant, I realize it's tall. But how did Mendel get to understand whether it was capital T or capital T small t? This is homozygous tall, this is heterozygous tall. How was he actually able to do that? Any idea? Do we get an idea? Well, the answer to this question is test cross. What do you mean by test cross? So in test cross, my dear children, what you do is, in a typical test cross, an organism showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent. Now, if I say this, you will not understand. So for that, I have to tell you some scenario. Suppose I have a condition where the plant can be this tall or this tall. To understand whether what kind of tall it is, what do I do is, I would be crossing it with the parent plant. Okay, with the parent plant, that is their parent, who is purely recessive, like this, homozygous recessive. Now you might think, ma'am, in that case, how will I able to understand? I don't get it. So I'll tell you. So suppose this is the parent, right? This is one parent, small t is one parent, and capital T suppose another parent. Agreed? And over here I have capital T small t as one parent and small t as another parent. Agreed? Okay, this can be one scenario. This is scenario 1, and this can be another scenario, scenario 2. So I just know the plant is tall, I do not know whether it's like this or this. For that, I crossed with the parent recessive plant. Recessive, okay, always recessive. So here the gametes of this plant will be small t, agreed? Here it will be always capital T. Agreed? Here the gametes will be capital T or it can be small t. Here it can be small t. I'm gonna cross. Suppose this gamete crosses with this, I get capital T small t. This crosses with this, again I get capital T small t. This crosses with this, again I get capital T small t. This crosses with this, I get capital T small t. Ma'am, all of them are tall. All of them are tall. They are heterozygous tall, but they are tall. Over here, when I cross them, what will I get? Let's see. So capital T, small t, capital T, small t, small t, small t, small t, small t. Ma'am, I got two plants which are dwarf and two plants which are tall. That means probably the plant that I had, if when crossed with this particular older plant, which is recessive parent plant, my dear children, if in the progeny all of them were tall, probably the plant that I was looking forward for might be having a genotype like this. If suppose the plant which was there, I crossed with this and I got that two of them were dwarf and two of them were tall, my dear children, in that case, the tall plant that I had a genotype like this. That's the use. The results, it would be drastically varying. And that is the use of test cross. So in a typical test cross, we'll read it once more, showing a dominant phenotype. A plant which is showing a dominant phenotype. Phenotype means physical character, dominant physical character, that is tallness. And whose genotype is yet to be determined. We do not know the genotype. It is crossed with the recessive parent instead of self-cross. Instead of doing a self-cross like this, my dear children. Capital T into capital T, capital D, if I do, my dear children, all of them will be tall only. Okay. And this if I do, I might not get the correct answer either way. So what do? I cross it with the recessive parent plant. Okay. In that, I know I have a fixed, one parent is at least fixed. I know that particular parent's character. So I can just understand the other ones. Okay, that's the purpose of it. So this is what you mean by test cross. And in your NCERT, there is a picture shown like this, where you are crossing between two plants. Okay, so there is a dominant phenotype, that is violet color. But I do not know if it is violet like this, capital W, or it is capital W, small w. So you might be thinking, ma'am, why is not VV? So it's sometimes like this also, it is usually put, but usually the dominant character's first letter has been taken. I don't know why they didn't, but anyways. So over here, you're unsure between capital W, and capital W, small w. So what do? You cross it with what? A white color plant, the recessive one. Okay, we learned, no, in flowers, the white is recessive. So we cross with that. And then we get to know, in that one case, all of them are violet. And in one case, 2 of them are white and 2 of them are violet. So probably this one, if the scenario was like this, then the violet color was capital W itself. Or the scenario was like this, then the plant having violet flowers, but they would be having a genotype capital W, small w. I hope it's clear. Children, always understand one very important thing. When you study principles of inheritance and variation, you might feel that it's a lot to study, there's too much. So in that case, what you have to do is slow down, pause the video, try to take things up, and then only go ahead. Okay? Now, why I have taken all the things together is that suppose we go ahead in the next week with midterm revision, I might not be able to cover this in Sankalp series. So I want you to have at least what I'm teaching it right now in a detailed way. That's the purpose of it. So if at all you're bored, please stay bored for a little more while, but please study. Okay, that's a challenge that we did to ourselves. Now there's a term that you will see, okay, that is test cross you studied right now. What do you mean by back cross? So crossing an F1 hybrid with either of its parent. So in test cross, what we are doing, we are basically crossing a dominant phenotype, organism having dominant phenotype with a recessive parent. For that, we are going back only. We are going back. So in back cross, proper back cross, we say whenever we cross F1 hybrid with either of the parents, it is known as back cross. It can be a dominant parent, can be recessive parent. So can I say this is a type of this? This is a type of this. Why? Because in back cross, anyways, you are going back and crossing with any of the parents. But now here you are specific. You are crossing just with one parent out there, and that is what? The recessive parent. So back cross means going back. Test cross means being specific about crossing only with the recessive parent. That's all. Do not bring too much confusion in your head. Okay? Clear? Done? Can we go ahead? So my dear children, until now, we were thinking about how whatever Gregor Mendel said was true. We spoke about his laws and things like that. Now it's time to talk about one very important thing out there, which is exceptions of Mendel's law. So when we said that, when we had the question like, was Mendel wrong? Okay, because we saw this, we saw that. In that sense, he should have been wrong, right? But the answer to this question is, he was not wrong. He experimented on green peas, and this is what he found. And it is usually true only. Whatever he said was completely true. Just that there were some exceptions to it. There can be an exceptional case everywhere, right? Similarly, Mendel also, with his experiments, he found that there were certain exceptions to what he said. He didn't find, someone else found, but anyways, there were certain exceptions, and we are going to talk about those exceptions right now. The first exception, okay, which is known as incomplete dominance, and the first example is that of snapdragon or antirrhinum species. So this is the name of the plant, please remember it, snapdragon, or I get confused with the pronunciation, antirrhinum species is what you're going to study now. What do you mean by incomplete dominance? You will study when I teach you that right over here. Okay, rather than me telling the entire definition, you study like this. So, I am telling you children that red color is a dominant color. and I'm telling you that the white color is what? The recessive color. So for white we should use W, no. The dominant color we can use that only, the same letters, okay? So R is recessive and this is what? This is white and over here this is red. I am giving you this information. And now we are going to cross, okay? So I had pure red plant, that means I had something like this, and pure white plant, that means I had something like this. These are what? The parents. Now in the gametes, what would be the gametes? The gametes can be capital R, it can be capital R, here it can be small r, here it can be smaller. Agreed? Now in the F1 generation, first generation, when I cross this and this I get capital R smaller, this and this I get capital R smaller, this and this I get capital R smaller, this and this again capital R smaller. Now in this condition, what is going to be the phenotype? Tell me. Physical appearance, what is it going to be? Ma'am, since R is dominant, it should be red. It should be red because all of it has it, right? But you know the twist in the story is what? It's not actually red, it will be pink in color. It will be pink in color. That means that's not according to what Mendel said, ma'am. It's not dominant. Yes, that's the entire thing. It's not dominant. There are certain alleles which are not completely dominant over the other allele. And this is what you mean by incomplete dominance. So capital R and small r, okay, over here capital R is not completely dominant over the small r, which is why you see a different color, red and white, the medium color which is there, the pink color appears out there. Okay. Okay, in that case, let's cross once more, no? Let's cross just like how Mendel did. Let's cross the progeny plants. Okay, so I'm taking the progeny plants over here, capital R, small r, capital R, small r. I want to see whether I can get more pink flowers. Have I completely lost the red flowers and the white flowers and things like that? So let's see that. So I took the parent plant, F2 generation parent plants, and now I have, F1 generation parent plants. Now I'm going to form the gametes. The gametes can be capital R or small r, or it can be capital R and small r. Agreed? Now the F2 generation, my dear children, I want to cross it. So this crosses with this, I get capital R, capital R. Agreed? This crosses, wait. It happens. Okay. This crosses with this, I get capital R, small r. And this crosses with this, I get capital R, small r. This crosses with this, I get small r, small r. Now, ma'am, the genotype, the phenotype. Bacha log, over here also I see that there are two conditions where capital R, small r is there. In this thing, the phenotype will be pink. Ma'am, what about this? Ma'am, what about this? There are only capital R's, right? In this condition, my dear children, it will be red. Because it's just there. The genotype, this only same allele are there, right? So that's why it will be still red. It's not dominating over something. Okay, both of them are same, which is why it will remain red. But over here there is someone else and it is not able to completely dominate over that, which is why it will turn pink. Now small r, my dear children, what will happen to this? This would be what? This would be white in color. So in the F1 generation you only see pink plants, but in F2 generation you see red, pink and white plants. And if you are asked about the ratio of it, if you are asked about the phenotypic ratio as in how does it look? So one is red, two of them are pink and one is white. Agreed? If you are asked about genotypic ratio, here comes a twist. Okay. Genotypic ratio in F2 generation of tall plants and dwarf plants were what? 1:2:1. The same thing is going to happen over here. Only the phenotypic ratio has changed, right? The phenotype there, it was 3. All the 3 of them were tall and 1 of them were dwarf. So it was 3:1. But here it is 1:2:1. In genotypic ratio, it still remains the same. This is one type of genotype, capital R, homozygous. Okay, capital R. And the second type of them is capital R, small r. And the third type of them is what? Small r. 1:2:1. Got it? So only the phenotypic ratio has changed over here. Why? Because it is showing incomplete dominance, and that is seen in snapdragon or anthrenium species. So this is the first example of incomplete dominance. There is one more example given in NCERT, not directly under incomplete dominance, it's given elsewhere, so I am going to teach you that also in this session. Okay, so please remember this is first exception. According to Mendel, he said in his law of dominance, one is dominant over the other one, that will express. But this is not the correct case. One can be incompletely dominant as well. Cool? Done? Snapdragon, anthrenium species, will you remember it? I suppose yes. My dear children, if you think that I am explaining well, what do you have to do? You have to tell me that. If you don't tell me that, will I come to know? No. If you don't do the homework, will I come to know? No. Okay, if you don't ask me doubts, will it be cleared? No. If you don't give me feedback, will I understand? No. So please do all of these things. It's very, very, very important. Now, so the entire thing is given in your NCERT also. You took red plants and white plants. So them, instead of writing the whole, what do you call, just a second, I'll show you. Them, instead of putting capital R in one, capital R in another, small r in another, small r in another, they just put one thing, one gamete, capital R, small r. And then they crossed and said that all of them are pink in color. Now they crossed both of them, capital R, capital R, capital R, small r, with the help of Punnett square, and they found this. Okay, so phenotypic and genotypic ratio remains the same in incomplete dominance, very, very important. 1:2:1, 1:2:1. In your normal plants, according to Mendel, the phenotypic ratio is 3:1, and over here it will be 1:2:1. Depending on the plants, okay, depending on the, what you get. Okay, so incomplete dominance is a type of inheritance when neither allele is completely dominant over the other. As a result, the offspring show a blending of traits. Okay, neither of them are completely dominant over the other one, which is why you see a blending of the trait, and that is about it. That is the first exception that we are having, incomplete dominance. Now going ahead with the next option, that is, exception, that is multiple allelism. According to Mendel, he said that capital T, small t, these are the two alleles of height, there is no other alleles. It can be just like that, dominant allele, recessive allele, that's all. But over here in blood group, there are three alleles, IA, IB, IO. Are they an example of multiple allelism? The answer is yes. When there are multiple alleles governing the same thing, that is your blood group, my dear children, or a character, you call this condition as multiple allelism. But you will not understand anything if I teach like this. So I have to give you an example. And when I give you an example, we go ahead with the next exception out there, which is known as codominance, for which the same example is being used, that is the blood group. So multiple allelism means when you have three different alleles or multiple alleles for one particular character itself, that is here blood group. Okay, it can be something else also. And the blood group shows one more exception, and that is codominance. It might be confusing for you as of now, but listen to me carefully. I am going to take all the confusions away from you. Okay. So I'll tell you one very important thing. When you talk about the red blood cells, your RBCs, on the plasma membrane, they have some sugar polymers, okay, that protrude from the surface. And the kind of sugar that should be there is controlled by a gene. It is controlled by a gene. Now this gene, my dear children, has three different types of alleles. One is IA, the other one is IB, and the third one is I, or you can say IO, okay, or you can say IO, is the same thing. So there are three different alleles controlling the same gene. This gene is controlling what? This gene is controlling the type of sugar which will be protruding from the cell's surface. Okay. So when IA, IB are present together, they both express their own. Okay, this I should be teaching you. So let's come and study about the blood groups, and it's really, really fun. Okay, so let's see. So over here, my dear children, suppose you have three different alleles, IA, IB, and IO, or you also call this as I. I can have three conditions. So over here, suppose a parent has IA, another parent, from another parent I am getting IA itself. In this particular case, what would be my blood group? If from one parent I am getting IA gene, from another parent also I am getting IA gene, what will be my blood group? My blood group will be A group. Okay. Suppose from one parent I am getting A, okay, and from other parent I am getting O. In this condition, what will be my blood group? Ma'am, I do not know. The answer is A is dominant over O, which is why it will be A itself. Okay, since I have this allele. Now, suppose I have A, from one parent it's A, from other parent it is B. In that condition, ma'am, In that condition, what will be the blood group? It should be A, right ma'am? A, I have seen it's dominant over O, so it should be dominant over B also. No, there's a twist. Both of them are equally dominant. Both of them are equally dominant. So this is known as codominance, which is why both of them will be expressing itself. It will be AB blood group. The same thing happens over here. Suppose from one parent I get B, from other parent also I get IB. What will be the blood group? The blood group will be B. From one parent I have IB, from one parent I have IO. What will be the blood group? Ma'am, I saw that O was recessive over here, but I don't know about here. It is still recessive, my dear children. It will be B blood group itself. Now suppose from one parent it's O, from other parent, from one parent it's B, from other parent it's A. What will the blood group? Both of them are codominant, ma'am. It will be AB blood group. Okay, it will be AB blood group. Now talking about IO. Suppose from one parent I get IO, from other parent also I get IO. In this condition, what is my blood group? Only in this condition, the blood group will be O. If from one parent I get IO, from other parent I get IA, here this is there to dominate over this. This will be A blood group. From one parent I get O, from other parent I get B. What's my blood group? It will be B because this is there to dominate over it. So only in one condition, my dear children, you get O, and that is this condition, IO. Okay, and in and in this condition where you get A and B, you get AB blood group. Fine. I hope you're getting this. So my dear children, over here you see multiple alleles are governing a factor, that is what? Your blood group, which is why this is an example of multiple allelism. And also this is an example of codominance, because when A and B come together, both of them are equally dominant, which is why you get AB blood group. AB blood group. If you want to take a screenshot of this, you can take a screenshot, and I'm going to tell you how many genotypes are there actually in the case of blood groups. So this is the possible things that I have told you, okay? When A crosses with whatever thing is there, and B crosses with whatever thing is there, and O crosses with whatever thing is there. But I'm going to tell you when you have, okay, I can put it here itself. I'll try to put it here. So when all do you get A blood group? You get A blood group when it is IA, and also when it is IA and IO. Only in these two conditions, okay? When do you get B blood group? When it is IB, or when it is IB and IO. Okay, in these two conditions you will get B blood group because this is dominant over here, A is dominant over there. Now when do you get O blood group? When there is IO and the other one is also IO. In this only condition you will get O blood group. When will you get AB blood group? When it is IA and when it is IB, you get AB blood group because both of them are codominant. So how many genotypes are possible in blood groups? 1, 2, 3, 4, 5, 6. 6 genotypes are possible in blood groups. Please remember that. 6 genotypes are possible in blood group. How many phenotypes are possible? So here it's A, here it's B, here it's O, here it's AB. These are the phenotypes. This is the thing. Blood groups can be only this much. So 4 phenotypes are possible. 4 phenotypes and 6 genotypes are possible in the case of blood group. And this is an example of codominance as well as multiple allelism. I hope you got it. Okay. So over here in your NCERT textbook, this is the thing that is given. The genotype of the offsprings, allele from parent 1, allele from parent 2. So I have just made it a bit proper over here. Allele from parent 1, allele from, so I'll write it like that. Parent 1 and parent 2. Parent 1 and parent 2. Parent 1 and parent 2. Okay. Now suppose, let me make you use your brain. What is your blood group? What is your blood group? I suppose, let's for example me. My blood group is A. My blood group is A. So what is the possibilities of my parents? What could be my blood group of the parents? Okay, what could be the blood groups of my parents? If my blood group is A, my parents' blood group, can I find that out? I can. Okay, I can. How? My dear children, for A blood group to come, the condition should be IA, or it can be IA and IO. Right? So over here, suppose my one of the parents had IA. And in the other thing it is also IA. Okay, this is parent 1 and suppose this is parent 2. So here it is IA This is parent 1 having both the alleles same. Here parent 2 also have both the alleles same. In this case, whatever cross I do, I'll get IA itself. So this can be one condition where both my parents are having A blood group. Both my parents are having A blood group. Second scenario, okay, this is the first scenario. Second scenario, it can be like one parent can have IAIO, other parent can also have IAIO. This is parent 1, this is parent 2. In this case also, parent's blood group is A itself. Here, this and this I cross, I get IA. My blood group can be A, right? This can be me. Okay, now if I cross this with this, I get IAIO. Again, since this is dominant, it can be A blood group. 50% of chances there that A blood group baby can be born. Now, let's see 50% or more than that. Again, if I cross this with this, IAIO. So the percentage has increased. So 75% chances there that the baby can be A blood group. Now this and this, IO crosses, my dear children, then it will be O blood group. 25% chances there that the child can be O blood group. So two people, just imagine, two people who are having A blood group, there are 25% chance that the baby can be O blood group. How? Because if this and this allele fuses, my dear children, you will get this. Okay, got it? So this can be a condition. Now, there can be more. Suppose the parent is like parent 1, parent 2. Parent 1 is IAIB, that means having AB blood group. Parent B is also having IAIB, that means having AB blood group. In that condition also, suppose this and this comes together, IAIA, that means A blood group, the child can be A. If this and this comes together, then it is IAIB, that means AB blood group. This and this comes together, again IAIB, AB blood group. This and this comes together, IB, that means B blood group. So 25% chance to be A blood group, 25% chance to be B blood group, 50% chance to be AB blood group. Get it? Okay, so my parents can be AB blood group as well. In that condition also, I can be A. There are so many scenarios, my dear children, or probably your one parent is AB and the other parent is just A. In that condition also, you will get. So many scenarios will be there. So you have to tell me what is your blood group and what is your parents' blood group, and you did try it up. If you are able to know your blood group and your parents' blood group, you will be able to understand which is the exact scenario which is there. So my parents, my one parent is A blood group, okay. My one parent has A blood group and my another parent has AB blood group. AB blood group. So in that case, my mother's genotype is clearly fixed. It will be IAIB. My father can be IA and IO, or he can be IA and IA also. IAIO, or it can be IA also. In either case, if I have one A over here, one A over here, this and this comes together, I will be A. So you can similarly find it out. It's fun activity. You can do that if at all you understand things. If at all you understood these things. My dear children, is it fun? I find it very fun. I usually do it when I was teaching in offline centers. My dear children, I used to do it. It was really fun. So this is the third exception that you have from Mendel's law of Mendel's law of inheritance. Usually he said that there cannot be two of the genes which are codominant, but over here you found that two of the genes were actually codominant. Okay, that is the third exception that you are having. So first one, incomplete dominance. Second one, multiple allelism. Third one, codominance. Over here, please remember these genes code for the sugar on the surface of the plasma membrane. Fine. Now, going ahead and studying the fourth exception, which is not written as fourth exception, but given elsewhere in your NCERT. That is polygeny. And over here, I have shown you a picture of so many people out there. Do you have any guesses why I have shown this particular picture? It's because polygeny is responsible for the different color that you have in your skin. Your skin coloration, why is too different? Why mine and yours does not match? Or whatever is there. So do you have any guesses? What does that particular term means, polygeny? It's very simple. Okay, you might be thinking that this is simple, ma'am. Yes. So usually what do we say? That a trait which is there, it is controlled by a gene. Height is controlled by just one gene according to him, according to Mendel. But over here, my dear children, they are saying that multiple genes on multiple chromosomes, or probably multiple genes on the same chromosome also, can be responsible for one particular trait. And that is over here, skin color. And we are the living example of that. So multiple genes comes together and codes for one particular character, that is what the skin color. And that's totally different from what Mendel said, because Mendel said one gene, one character. That's not the case. Multiple genes are there. I'm not talking about multiple alleles, not just multiple alleles, I'm taking multiple genes. Here one gene, here two genes, here three genes, all of them are coding for something, that is what your skin color. And I'm going to explain you right now about that. Suppose this is chromosome 1. Okay, from over here you have capital A, okay, these are the 3 chromosomes I have. Suppose I get from here one capital A, from here I get capital B. Okay, just a second. If you can think like this, if from both the parents I have got capital A, that means the genotype will be like this, right? If from both the parents I have got capital B, that means the genotype will be like this. If from both the parents I have got capital C, it will be like this. In this case, the skin color, my dear children, since all of them are dominant, it would be the darkest skin color that I would be having. Why? Because all the genes are contributing and all the genes have the dominant one over here, the dominant alleles over here. Suppose now the scenarios has changed. This is first scenario, okay? Now the scenario has changed. It is now capital A, small a. From one parent I got capital A, from another parent I have got small a. So here it will be capital A, small a. Here it will be capital B, and capital C is capital C. The skin color, if it was dark over here, it will be less dark. It will be comparatively less darker over here because I have got a recessive allele. Okay? So it has decreased. Okay, it has decreased. Now over here, suppose I'm going ahead making more changes. It's small, capital B, small b. From one parent I got capital B, from other parent I got small b. So now it has been like this: capital A, small a, capital B, small b, capital C, capital C. Again, it will be lighter version because now I have 2 recessive alleles, or I can say that the dominant alleles are less. Dominant alleles have become less. Now third scenario, my dear children, here it is small c and this is capital C. This is capital C, this is small c. So capital A, capital B, small b, capital C, small c. This again would be what? Lighter. The skin would be lighter. Now which one is the one where it will be the lightest? You think like that. Which one is the one where the skin color would be the lightest? Where all of them, my dear children, are small ones. So the scenario in this case will be AA, BB, CC. There is no dominant allele and this is the case where it will be the lightest. So how is this an exception from Mendel's law? Because Mendel said, according to Mendel, only one pair of alleles. Okay, it can be like this, it can be like this, either way. But only one pair of allele, capital A, small a. They were responsible for one particular character out there. But here it's not like this. There are multiple genes out there, multiple alleles out there, which is responsible for what one thing, that is your skin color. All of them are contributing, this protein, this protein, this protein, all of them are responsible for what? Your skin color out there. So this is an example of polygeny. Traits which are controlled by multiple genes are known as polygenic traits. Example, your skin color and also the human height which is there. So human height is a result of what? Polygyny. It's not incomplete dominance. Tall and dwarf, we get merged and that's how there's a blending and that's how you get it. No. There are multiple genes. Depending upon the product which is being formed, your height and your skin color is determined. Always remember these 2 examples are very, very important. Skin color and human height. Now each dominant allele, capital A, capital B, capital C, adds to the trait. That means it gives the dominancy. While the recessive allele small a, small b, small c reduce it. Okay, more dominant alleles result in darker skin, while more recessive alleles lead to what? Lighter skin, creating a continuous variation, which is why we have different colors, so many n number of color shades out there. I hope now you get it. Okay, and I hope in your school, if you didn't understand it, if you have understood it right here, you have to let me know that now polygyny is all clear, ma'am. Okay, so it's a difficult topic. I never understood, I never even remembered learning this in the school. Okay, which is why I'm telling you, it's not that easy to understand, but when you get the hold of it, when you have a little understanding of things, it's easy. So here multiple genes are involved. Polygeny, poly means many, and geny means genes. So polygyny, multiple genes, okay, multiple genes, write this somewhere. multiple genes, one trait. And the next thing that we are going to study is the exact opposite of it. Exact opposite of it. Where you will have one gene coding for multiple traits. That is also not something that we studied. One gene, capital T is for only tallness, no, nothing else. But here, according to the exceptions, my dear children, one gene sometimes can have what do you call effect on multiple traits out there. So over here, multiple genes, one trait, and over here, one gene, multiple traits. And I am going to explain you how. So my dear children, there is a gene, okay, there is a gene that codes for a very important enzyme, a protein, okay, that is phenylalanine hydroxylase. What is it? Phenylalanine hydroxylase. And what does it usually do? It converts phenylalanine, it's an amino acid, to something that is known as tyrosine. Phenylalanine to what? Tyrosine. That is the usual thing that it does. Okay, so it got stopped abruptly. Okay, my dear children, I was saying this, that suppose this guy is not there, this conversion won't happen, which is why the phenylalanine which you see over here gets accumulated in the form of phenylpyruvic acid. And when it gets accumulated, my dear children, what will happen? There are chances that it could lead to mental retardation. Okay, and what do you call this condition where there is a defect in the gene coding for phenylalanine hydroxylase? When do you see all this thing? This particular disorder is known as phenylketonuria. This is a genetic disorder. You will study about this later. So, I was talking about pleiotropy, saying that there is one gene controlling multiple traits. Ma'am, you didn't talk about the multiple traits until now. Yes, I didn't talk, and I am going to tell you now. Listen to me carefully. So, you think that this has just one function of converting this to tyrosine, right? But that's not the only thing. Now, tyrosine is being found. This tyrosine is very, very important for the production of melanin. And melanin is something that is responsible for our skin color also. So, tyrosine is not there. Melanin cannot be formed much. And in that condition, what will happen? In that condition, one more trait is going to get affected, and that is your skin and hair pigmentation. Skin and hair pigmentation. So one gene is not just controlling the formation of tyrosine, but also the next thing out there, which is skin and hair pigmentation, because tyrosine is needed for melanin production, and melanin production, my dear, when it's not there, what will you have? Your skin and hair get hypopigmented. You will not have too much pigmentation, it will get less. You will be what you call more fairer, more whitish in color. You get it? That's the thing that happens. So that is the meaning of pleiotropy, where one gene is coding for multiple traits, my dear children, and that's again an exception that you see. So according to your NCERT, there are just 3 exceptions given in the exception column, and that is incomplete dominance, there is multiple allelism, and there is codominance. But also understand, pleiotropy and polygyny are also examples of it. Remember the names. Phenylalanine is an amino acid. It converts to tyrosine with the help of phenylalanine hydroxylase. Okay, hydroxylase, there's an enzyme out there. If it's not happening, it gets accumulated in the form of phenylpyruvic acid. If this is getting accumulated, you get mental retardation on all of these things. This condition is known as phenylketonuria. Ma'am, the other trait which is getting affected is what? Skin and hair pigmentation. It gets hypopigmented, my dear children. So this is an example of pleiotropy. So when a single gene influences different metabolic pathways or phenotypes, always remember like that. A single gene is therefore multiple traits, or you call multiple phenotypes, physical appearance, right? So phenylketonuria is an example, or is caused by mutation in the phenylalanine hydroxylase gene. The phenylalanine hydroxylase enzyme converts phenylalanine into tyrosine, and in its absence, phenylalanine accumulates as phenylpyruvic acid, causing what? Mental retardation. The second character that it hampers is reduction in hair and skin pigmentation due to low tyrosine levels. That's all. Okay, that's all. I know it's too much to learn, but it will be easy. Okay, in the long run that you see, my dear children, when you read, it will be easy. Now, I have one example of both incomplete dominance and pleiotropy. I told you while talking about incomplete dominance, there is one more example, right? So, I want to discuss about that, and that example is also an example of pleiotropy. Okay, so my dear children, there is a genotype capital B, there is a genotype capital B and small b. Okay, so whenever this capital B comes in, the starch grain size would be large. Okay, the starch grain size would be large, and the seeds are round. Okay, the seeds would be round as well. So it is there for largeness and also for making it round. Small b when it comes, it will be small and it will be wrinkled. This is what? I'm talking about the size, and this is what I'm talking about the shape. Now when capital B, small b comes in, there is medium. Okay, there is what? Medium in the size. But if you talk about about the shape, the size is medium, but the shape, my dear children, since it's capital B small b, it will dominate. And what's the character of capital B? It's round. So this will be still round. So what do you understand from this, ma'am? I didn't understand anything. So for seed shape, if you talk about seed shape, my dear children, there is capital B which is dominant, and that wins. But for starch grain size, if you see incomplete dominance. Okay, you would be seeing incomplete dominance. Why? Capital B is not completely dominant over small b, which is why you are not able to see large size. You will be seeing medium size out there. So this is an example of what? Incomplete dominance. When you talk in respect of the size of the seed, when you talk about the shape, it's showing complete dominance. When you talk about the size, it shows incomplete dominance. And how is this an example of pleiotropy? One particular gene which is out there, okay, one particular group of genes, capital B, small b, whatever is there, it is controlling two characters at once, two traits or two phenotypes at once. One is size, another one is shape, which is why this is also an example of pleiotropy. It's not given any NCERT that is given example of pleiotropy, but I am telling you it's an example of pleiotropy. Now where do you find this? You find this after your blood group and things like that, no? When it ends, there is a paragraph where all of this is written. And I wanted to tell, I do not want you to miss out anything, that is why I told you. So when it comes like this, always remember, in the case of size, it shows incomplete dominance. In the case of shape, it shows complete dominance. That's all. Okay, that's an example. With that, my dear children, we complete everything up there, and now I have a I have a homework question for you. I always tell, I give homework question when I feel that you can do the question, or when I think that I have taught you and you can easily tackle it. Now I forgot the number of questions that I gave you, so I think it was homework number 3, right? So this is homework number 4. Find the correct answer. Generally, a gene regulates a trait, but sometimes one gene has effect on multiple traits. The trait AB blood group of man is regulated by one dominant allele and another recessive allele. Hence, it is co-dominant. You have to tell me what is your answer. Both the statements are wrong. Statement 1 is correct. Statement 2 is correct. Both the statements are correct. Either way. Okay, this is homework number question 4. Homework question number 4. Okay, please answer that. Now going ahead with the question that I will be doing for you, that is, the genotype of a husband and wife are IAIB and IAIO. This is one husband, this is husbands and this is wives. Among the blood types of the children, how many different genotypes and phenotypes are possible? This type of questions come. This is why I have put down over here. So one parent is IAIB, right? The other parent is IAIO. How will you find out the possible genotypes and phenotypes? Let's see. So in gametes, it can be, okay, in gametes always write like this, okay? And over here, IO, right? This is B, this is A, this is O, and this is A. This and this crosses, I get IA, IA. Agreed? This and this crosses, I get IA, IO. This and this crosses, I get IA, IB. And this crosses, what will I get? I get IB and IO. What are the possible phenotypes? Let's check that. Physical appearance. This is A blood group because both of them are A. This is again A blood group because A is dominant. This will be a B blood group and this will be B is dominant, so B blood group. So how many phenotypes are possible? Ma'am, 1, 2, 3, 4. Wrong. Both of them are showing A only, no? So 1. This is considered as 1. A, B and B. 3 phenotypes are possible. 3 physical appearances possible. What about the genotypes? I see all of them are different. 3 phenotypes, let's find. 3 phenotypes, yeah. 3 phenotypes are there and 4 genotypes, IAIA, IAIO, IAIB, IBIO. 4 genotypes are possible. So 4 genotypes and 3 phenotypes, your answer would be B, my dear children. Okay, this type of question if it comes, will you be able to do it? Yes, my champions would be able to do it. Now the next question, ABO blood type in man is an example of pleiotropy, incomplete dominance, codominance, multiple allelism. What is your answer? Does it show pleiotropy? No. One gene, one trait, that is the thing that it shows. Incomplete dominance? No. Codominance, yes, A and B shows codominance. Multiple allelism? Yes, they also show multiple allelism. You have 3 multiple alleles. So can I say C and D? C and D only. Your answer is option C. Cool. Okay. Now choose the possible genotype responsible for the lightest skin color in human beings. This came in 2018. Tell me. Lightest skin color. When all of them are recessive, that will be lightest. So small a, small b, small c, small c. This is your answer. Now do you feel confident that, oh my god, this is how we solve question? Things like that. Now I think I am ending the session with homework number 5. There are so many homeworks for you today, right? Why? Because this needs practice. If I teach you and I go, you will forget it. So what is a possible blood group of a children whose parents are heterozygous for A and B blood group? Both the parents are heterozygous for A blood group and B blood group. What are the possible blood groups of the children? You have to cross it and find it. Remember, it's heterozygous given for both A and B. You have these options. Tell me the answer in the next section. Now I hope you are able to understand whatever I try to teach you. It might have taken a bit longer, but my dear children, understand the importance of this chapter, and I understand how it is important for you as well for scoring great marks, which is why I took a lot of time to explain this to you. If at all you felt that the teaching was good, if at all you felt that class was nice, you understood things, I won't say it was fun, and I don't know whether it was fun, because obviously you talk about serious stuff when you talk about all these big things. But anyways, I tried my best. You let me know if at all you liked it. Do not forget to share it with your friends right now. Okay, we have to share it with our friends, and also I hope you have finished your challenge. If you have done, kudos to you. All the best, wishing you all the best, and my dear children, very proud of you. Keep working hard. Lots and lots of love from Jasmine Ma'am. Bye bye, take care. Do not forget to subscribe to the channel if you have not subscribed it yet.